Folks, this video is a response to a comment left on my video, Richard Sternberg, Endogenous Retroviruses Debunk. Now, after providing the information in the video itself that refutes the claim of endogenous retroviruses being evidence of evolution of mankind and man having a common ancestor with an ape-like creature and a relationship, therefore, to chimpanzees, this fellow here leaves this comment where he makes a number of points, all pretty much regurgitating the same thing, still claiming that ERVs are evidence of common ancestry, even though the video that he's commenting on refutes the idea. It's as though this information in this video just went in one ear and out the other. But I'm going to debunk what he says. Barry, I'm going to refute your argument here for you in this video, because you seem so dead certain that even though the information provided in this video refutes your idea of endogenous retroviruses, you went ahead and posted this long post on my video here, still claiming that ERVs are evidence of common ancestry. First, a little bit about ERVs, so that people who don't know what this subject is about will understand. An ERV is called an endogenous retrovirus. That's what it's called. The name is a misnomer. There are no such thing as retroviruses. What is believed to be an endogenous retrovirus is supposedly it is the DNA of a virus that gets inserted into the DNA of a common ancestor of man, with chimpanzees in the past, before human beings and chimpanzees evolved from their common ancestor. So the animal which was the common ancestor between human beings and chimpanzees gets a virus, its DNA gets inserted into its DNA. Then, because human beings and chimpanzees both evolved from that common ancestor, we both carry this viral DNA in our genome. That's the claim. I'll restate it. Maybe I didn't state it clearly enough. Let's imagine in the imagination of the evolutionist, man has a common ancestor with the chimpanzee when we go far enough back in time. There's a creature from which the evolutionists believe both humans and chimpanzees came over time. This creature diverges. New types of creatures arise from this common ancestor, and over time you get chimpanzees and human beings both evolve from this organism that was once a common ancestor of both human beings and chimpanzees. Now, in the DNA of that organism from which humans and chimpanzees arose, supposedly, there is a virus that gets the organism gets an infection from a virus. The virus's DNA gets inserted into the DNA of that organism. Then, because human beings and chimpanzees evolved from that common ancestor, we both carry the same sequence of viral DNA in our genomes, and this is supposedly evidence that we're related to chimpanzees by a common ancestor, you see, because we both carry the same viral DNA that was inserted into the DNA of the animal from which both came. Understand? The problem is this. This claim is largely based on the old idea of junk DNA, which is now thoroughly dismissed. So let's look at your claim. You say, what is the case for common descent from ERVs? You say, firstly, Barry, all you're doing is describing what you believe is the process. Okay, this is not the evidence. You're describing what you believe took place. Retroviruses replicate, etc. Okay, viruses get into a cell and they replicate. Right, we know that. Retroviruses tend to target certain types of cells. Okay, no argument there. The insertion is made by a retroviral enzyme called integrase. Okay, so we know the viral DNA can get inserted into the DNA. That's true. We find in genomes of creatures such as ourselves and chimpanzees inherited structures that appear to be broken retroviruses insertions. That's your first mistake right there. That's where you make your first mistake. I'm going to explain that. Because they find the same sequence of information in the chimpanzee genome and the human, they believe that it's a viral DNA that's now shared by both. Okay, this is what I'm going to refute for you. Then you say, although certain components from some ERVs perform functions in the host. Certain? That's misleading. Certain. It appears that all of them do. Not certain. So what you've done here by using the word certain is attempted to play down the functionality that has been found in these supposed ERVs. We have discovered a lot of very critical, important functions for these what are called ERVs, sequences of information in human and chimpanzee DNA. They are not functionless junk. That's important. Keep that in mind. Okay, so you acknowledge there are functions to them, but you attempt to downplay that by saying certain components of some ERVs. It's very misleading, Barry. As you'll see, very misleading. It is believed that they all have function, and the evidence shows that they all do. In fact, have critical function. You say one or two of them being essential in some species. Very, very wrong, Barry. Very, very wrong, Barry. That's wrong. Very wrong. I don't know where you got your information, but it's grossly outdated, or you're just attempting to be dishonest. One or two even being essential in some species. No, Barry. Critical function in all of them. Okay. So, then you say, the only explanation that makes sense is the ERVs are the result of retroviral insertions. No, Barry. That's not true. This is not the only explanation. The only explanation is not that a common ancestor with human beings and chimpanzees got an viral infection, and this is why we share the same sequence of information in our genome, and that it has to be a viral insertion. That's not. That is not the only explanation, Barry. I don't know how well you seem to. You apparently do not research this subject very well, and you've swallowed hook, line, and sinker the claim of the evolutionist without research. You should investigate the claims made from both sides and then go to the data to see which one is right. See, that's how I became a young Earth creationist. By reading the claims of the evolutionist, reading the claims of the creationist about the same thing, and then going to the data to see what the data actually says and see whose argument fits the data. That's the way I got out my mind, broken out of, freed from evolutionism, and became a young Earth creationist. By comparing both camps' arguments on the subject, then going to the data to see whose argument fits the data. You have not done that. You have not done that. You say all human beings have some 200,000 plus ERV fragments. We share a lot of bits of information, it seems, same code with many other creatures, including chimpanzees. And here you repeat the same thing. Okay, so now, Barry, here is the refutation to your claim. Firstly, the claim of ERV is made based largely on the junk DNA idea. You see, it's been claimed since for about 50 years now that the majority of the DNA is junk, garble, messed up garble left behind by evolution in our genome. And that only about 3 or 4% of our DNA is actually functional and useful. That is the stupidest idea. It was an idea made when our understanding of genetics was in its birth, going through its birth. We know so much more now, and we still have so much more to learn. We still only know a fraction of what there is to know about DNA or the cell, how it functions. But the junk DNA claim was made long ago, 1950s and 60s. It was promoted. And what we have discovered since then, is that the cell, the parts of the DNA of a human being, which seemed to have function, accounts for about 3 to 4% of the DNA. So the evolutionists assumed that the rest of it is junk. That 3 or 4% is what codes for proteins. What we've learned since then is a lot. What we've learned is the rest of that DNA has function too. Just because it does not code for protein does not mean it's junk. Since then, the Human Genome Project and the ENCODE Project have discovered that 98% of the human DNA is now believed to be transcribed by the cell. In other words, the information is lifted off of it and read and made into RNAs. An RNA is a string of information that's used to regulate gene expression. It makes proteins, does various things. And now we know that the vast majority of the entire DNA molecule is transcribed by the cell to have function in the cell. It's functional. So the junk DNA paradigm is out the window. And these ERVs existed, most of them exist, in what has been called for 50 years by the evolutionists, junk DNA. Now, let me point out this about the junk DNA claim. It was very damaging to science, as all evolution claims are. They hold back science. I'll explain why. The idea of the evolutionists that the majority of our DNA was junk, garble left over by evolution, did damage to genetics research for 40-something years. Because they believed that it was garble, they didn't spend a nickel to investigate it until relatively recently, holding back genetics knowledge. Had the evolutionists' idea that the majority of our DNA was not junk not stood in the way of genetics, more of it would have been studied, and we'd know a lot more about DNA today than we do, thanks to the evolutionists. So evolutionism, that's a beautiful example of how evolutionism has held back science by claiming the belief that the majority of our DNA was non-coding garble junk, had no function, and left behind by evolution, and only 3% or so of our DNA was actually functional, held back genetics research for 40 years. Thank you, evolutionists. You see, the evolutionist paradigm, their false ideas got in the way of real science because they believed a false thing about DNA. It's now known today that it's believed 98%, and this is from the secular camp, from the white papers in the journals. Even the evolutionists today are acknowledging that 98% of our genome is transcribed by the cell because it's functional. It's not junk after all. It's critical, in fact. Some of that DNA has been observed to code for proteins. Other portions of it, a large portion of it, it seems, is necessary for embryonic development. And in fact, these ERVs, here I'm getting to the real point of it. Many of these ERV sequences that have been studied, that are sequence of information found in the genome of a human being and in the chimpanzee, because they're the same looking, they're believed to be viral DNA that was inserted in our common ancestors' genome before humans and chimpanzees evolved. That idea has been shot to heck. Because we now know, the more we study these, the more that we discover they have function that is critical to the existence of the organism. In fact, your organs and the human placenta as well are now known to be regulated. The gene expression that produces those during embryonic development is regulated by what are claimed to be endogenous retrovirus sequences. See, it's not just functional, it has critical function to the organism. It's information necessary for a human being to exist, or a chimpanzee to exist. And one problem for this is this. 80% of the proteins between humans and chimpanzees are different by at least one amino acid. That kills evolution theory right there. And now, if we find an ERV in a chimpanzee and in a human being, and we know that they are necessary for the development of the organs of a human being during embryonic development, it's likely that's the case for the chimpanzee also. It's critical information for them as well. But the fact that we have 80% of our proteins are different from theirs by at least one amino acid demonstrates that we have a biochemistry that is so different that to believe that we are related by evolution is nonsense. Since the evolution of all these proteins in the organism cannot have taken place because it would not yield a viable organism. When a mutation changes a DNA, the sequence of information that codes for a protein, it makes a malformed protein, which is either completely functionless and then chopped up by the cell and reused as food to make new proteins, or it has reduced function, and this is the cause of diseases like Alzheimer's and Tay-Sachs. So you see, the biochemical difference in the biochemistry between human and chimpanzee is so great, evolution cannot be true. But moreover, these what are called ERV sequences, since they were now known, many of them, to be critical to the development of the organs of a human being and therefore also a chimpanzee during embryonic development, because our biochemistry is so radically different. Remember, 80% of our proteins are different by at least one amino acid. And one amino acid is critical. It changes the function or makes it completely dysfunctional, reduces functionality, or makes it dysfunctional when a mutation changes an amino acid sequence by even one. Even one. It's so specific. A protein is so specific a machine, microscopic machine, that a change of a single amino acid in most proteins will make it either dysfunctional or reduced in function and may cause disease. One. So the evolution of human beings and chimpanzees from a common ancestor is illogical and not possible, since it cannot be said that all of these proteins would evolve. See, proteins can't evolve. That's the thing. And they would have had to for human beings to be related to chimpanzees. That kills evolution theory. It's dead. No go. Now, because these ERVs are critical to the function of the development in the embryonic stages of the organs of a chimpanzee and a human being, and because the biochemistry of the chimpanzee and the human being is so radically different based on protein alone, this information is therefore not related between a human and a chimpanzee. Not by common ancestry. It's merely code that has a similar function. Now I'll explain that. Let me show you. This is from Conley et al. We report that the existence of 51,197 ERV-derived promoter sequences that initiate transcription within the human genome, including 1,743 cases where transcription is initiated from ERV sequences that are located in the gene proximal promoter or 5 untranslated region. Also, our analysis reveals that retroviral sequences in the human genome encode for tens of thousands of active promoters. Transcribed ERV sequences correspond to 1.16% of the human genome, and PET tags, capture transcripts initiate from ERVs, cover 22.4%. Wow. Of the genome. Wow. 22.4%. Now, it's illogical also to consider that the information in the little DNA of a virus could have the critical information necessary to regulate the gene expression of thousands of human genes so as to produce the liver, the pancreas, the kidneys, etc., and the brain. Is that really logical? Of course not. How in the world is it logical to believe that a little string of information in the DNA of a virus has the power, the specific code necessary to regulate many human genes in order to produce organs that are vastly more complex than the virus itself? Think about that. See, that's not logical at all. Not logical. That's like believing that you can have a one paragraph sentence of instructions that somehow possesses the remarkable information necessary to regulate teams of builders who will assemble a space shuttle. Is it logical to believe that you can squeeze into the one half a paragraph long sequence of instructions related to the assembly of a bicycle can somehow regulate the workers to assemble thousands of different parts to assemble a space shuttle. That's not logical, is it? Neither is it logical to believe that the little snippet of information from a virus could have the information necessary to regulate the thousands of genes that produce the kidney, liver, brain of a human being and the placenta. Is that logical? Of course it's not. But evolution is not logical. Never was. It's not scientific. It's not science. Evolutionists believe the most outrageous things because they're willing to, because creation can't be true in their mind. So you see what this information is showing us here is that these ERVs have critical function related to human embryonic development. It's also known to be so in mice, etc. They are transcription promoters. See, where they say transcription is initiated from ERV sequences. See, in other words, these are regulatory functions. Regulatory. Active promoters, here they call them. Our analysis reveals that retroviral sequences, what they are called, in the human genome encode tens of thousands of active promoters. Now, if the human being, if what is called an ERV sequence is actually the information from a virus that was inserted in a common ancestor of chimps and man, how did it get the information to regulate the genes to produce the organs in an organism that is vastly superior in complexity to the little virus? You see, that makes no sense. Doesn't make any sense at all. Moreover, the organism needs if the organism that that the retrovirus gets inserted into already has information necessary to promote the active to the thousands of transcription factors necessary to regulate the genes to produce the organs of that organism, what are we believing here? Are we believing that the organism, the chimpanzee and the human being, spontaneously poofed into existence, waiting for the retroviral information to get in their genome so that they could produce the organs, which are apparently already encoded with vastly more complex information than is found in the genome of a virus? If the existence of the organism, the organs, in the chimpanzee and in the human being rely upon retroviral sequences, how did they exist in the beginning without them? Do you get that? We already have them. You see, these organisms already have these various organs, a brain, placentas, kidneys, liver, pancreas, whatnot. Now, if you believe that the retroviral information, which now is known to be what's called retroviral, is necessary to promote the gene functions necessary to produce those organisms, those organs, how in the world did the organs exist before the DNA, the retroviral sequences got in there? Do you see the problem? It means ERVs cannot be retroviruses because they have these functions of regulating genes so that to produce the organs of the organism. I don't know if you understood what I just said. I'll try. You probably did get it, but I'll just say it again, maybe so it'll really sink in. What are called retroviruses are known to have function in expressing, regulating the genes that produce the organs in a human being, in a mouse, etc. Now, if those organs if they are dependent upon what are called retroviral sequences in order for them to be produced, how did they exist before the DNA of the virus got into the genome? You see what I mean? This tells us they are not retroviral sequences. I'll explain to you what they are in just a minute. So this information just kills the evolutionists' idea. Now, first, the false claim of junk DNA, we know this is not junk. Secondly, we now know that they have critical function in the development of the organism. throws the ERV question out the window immediately. It's done. But regarding the junk DNA claim, this is what Dr. Maddock, director of the Institute of Molecular Bioscience at the University of Queensland, Brisbane, Australia, has to say about the evolutionists' claim of junk DNA that they've pushed forward for 40-something years. The failure to recognize the full implications of this, particularly the possibility that the intervening non-coding sequences may be transmitting parallel information, may go down as the biggest mistake in the history of molecular biology. And also, he says, the molecular taxonomists who have been drawing up evolutionary histories or phylogenies for nearly every kind of life are going to have to undo all their years of junk DNA-based historical reconstructions and wait for the full implications to emerge before they try again. In other words, all of the evolutionists' retroviral claims, junk DNA claims, and phylogeny, that is, their belief that because of genetic similarities between various creatures in this world is evidence that they all are related on some Darwinian tree of life, is out the window now. It's all been shot to pieces by modern science. You see, evolution theory is an old idea. It was packaged into a pseudoscientific theory by Charles Darwin long before anybody knew anything about biology. In Charlie Darwin's day, they didn't know squat about biology. They did not even know that DNA existed. Charles Darwin's book was published years before the discovery of DNA. So evolution theory has run into modern science and got squished like an invading bug. So, Barry, because these ERV sequences, what are called ERV sequences, are now known to have critical function in the development of organs, and the placenta and the brain and the heart and the liver, the kidneys, the pancreas, all this, they cannot be retrovirus. So what are they? I'll show you what they are. This is from w3schools.com, the world's largest web developer website. Here we see a little snippet of code. It says, window open. You can use this code in Java. You can use it in Microsoft C++, Borland C++ programming. Use it to close a window. Here's close window. There's open window. You can use blank URL, parent, self. These are tags, top, name. Full screen, yes or no, channel mode, directories, menu bar, height, left, resizable, scroll bar, status, toolbar, width, top, etc. These are all snippets of code. You can find them in your browser when you click File, Open. You'll find them in Microsoft Paint when you click File, Open. You'll find them in the Borland C++ programming environment software itself. Click File, Open. File, Close, Exit, Sort, List, Find. These sequences of information can be much more longer than this. You could have a string of a dozen commands altogether, or 2 dozen. And they do the same kind of thing in one application as they do in a different one. And that's what an ERV is. It's a snippet of code that God used in both the chimpanzee and in the human being. And if there was a common, and whatever organism that the evolutionists believe was a common ancestor to us, a lemur-type organism, whatever that was, that one too. And in an elephant, and in a wasp, and in a giraffe, and in a tarantula. You'll find these same snippets of code because they have function, because they're both biological organisms, and they both have to transcribe DNA, they both have to produce protein, they both have to deal with potassium, they both have to deal with iron and manganese. And you're going to have various functions. Read this frame, copy it. That's going to be found in the genome of every living thing on the planet. So what are they? They're snippets of code that can be found just like snippets of code, useful in one more Microsoft application or another, God did the same thing. He used snippets of good code because they're necessary to create an open window in a Microsoft program, or they're necessary for reading the frame of information on the DNA molecule. They're not evidence of common ancestry, they're evidence of a common designer. Now, how do we know that? Because they have critical function relating to the expression of genes that develop the organ of an organism during its embryonic development, and therefore cannot be retroviruses. That cannot be viral information that got inserted into the genome of a common ancestor with human beings and chimpanzees, and then when they split it off, they both carried it with them. They cannot be the information of a virus, because the existence of the organs cannot wait for the information of a virus to get inserted. They already exist. Do you see? They cannot be retroviruses. They're not viral information at all. They're snippets of code used by the common designer, by God Almighty. Okay? Now, just for fun, you might want to read this by Sean Doyle, Large-Scale Function for Endogenous Retroviruses. This is a very interesting article, and you can find many more like it. Now back to the video that you attempted to debunk, which debunked you before you attempted to debunk it. Microbiologist, evolutionary biologist Richard Sternberg shoots down a claim of ERVs several years ago amongst his peers, and you can hear him do so, and put his own peers to shame. Now, Barry, I hope you learned something here. Do you understand now how these supposed ERVs cannot be the information from a virus? Because they have critical function in the development of the organism during embryonic development. And the organs of the organism can't sit around waiting for the ERV to get there, because they already exist. Enjoy.
Yeah, I want to respond on the retroviral sequences topic. He's absolutely right. There are hundreds of thousands of copies of these virus-like sequences in our chromosomes of mammals and fishes and even the yeast cell. Now, what's interesting is that he's wrong on a number of points. Here's the first point. One of the first class of sequences to be expressed shortly after the first or two-cell embryo, that is from the zygote, including our zygotes, mouse zygote, to the two-cell stage embryo, first class of sequences are the endogenous retroviruses. And you know, it's determined in 2004 is that if you knock those sequences out, you block their expression, you stop embryogenesis at the 4-cell stage. Actually, it's cited in a paper that Jim Shapiro and I, one of the papers published in 2005, where we dealt with this topic, this whole issue of why this so-called junk DNA is functional. Here's another example. What holds the placenta together is a class of proteins, and they're essentially, they form tethers. And the proteins that are coded are specified by retroviral-like sequences. It's different in the mouse than the ones in the human. If you look now, there are these switches that flip gene sequences on and off. They're called promoters. Like you can think of them as molecular toggle switches. A recent analysis of the number of these genetic switches in our genome contributed by these endogenous retroviruses, so many of them they just have to be garbage, 181,000 roughly. So, and you look at the literature, what they're doing now, and this is, the ENCODE project was published in 2007, a series of papers where they looked at not just the sequences, but the expression of sequences, how they're copied in RNA. And what they found, to their surprise, is that most of the expression is taking place in this so-called non-coding DNA. And you can find papers in Nature and Science, genome research, genome biology. Thousands of them are out there now. Go to PubMed.org again, just type in junk DNA and see what you pull up. Don't take my word for it. But what you find is that when they investigate these classes of sequences, as numerous as they are, they're finding a whole series of functions. Pseudogenes, for example, so-called pseudogenes, their expression regulates, yes, they don't code for proteins, that's true. Do they look like defunct copies of other genes? Yeah, they do. But you have two papers back-to-back in Nature, and what they demonstrated is that, well, those so-called non-functional RNAs, the transcripts, are regulating other genes. And I'll be happy to provide you with the PDFs and with the references for those papers.
Okay. I have a comprehensive list on page 407 of my book.
Well, there you go, my friend. Barry, what you call ERVs are not retroviral information inserted in the DNA of a common ancestor by a virus. They're critical snippets of information, just like Windows codes, that one programmer uses to write a window, to say, create window, open window, search for this, copy and paste here, make this, go to this line and perform this function. That's what they are. They cannot be retroviral information from a genome of a virus because they have critical function in the development of the organism. And the organs of the organism cannot sit around waiting for the ERV to get there because they already exist. Well, there you go, Barry. I hope you enjoyed learning something today. God bless you.